What does a coal-fired power plant, a tuna, and a vaccine have in common? It may sound like the beginning of a bad joke, but the answer is serious. All three contain mercury, but as it turns out, not the same kind of mercury. Mercury is actually ubiquitous, present in the air, water, and soil all around us. Even before industrialization began, and even in remote regions far from human activity, mercury can be found in concentrations measured in nanograms per cubic meter. So simply saying that mercury is found in a sample doesn't provide any context. And not all forms of mercury are the same. So what are these different forms? Well, mercury can get bound up in several chemically different compounds, and those compounds behave very differently once they're inside a living body. So the useful question isn't, is there mercury in it? The useful question is, which forms of mercury are present, in what amount, and how does the body handle it? Today, we'll trace mercury through those three sources, look at how researchers measure what it does, and work through the kinds of questions that let you evaluate the science for yourself. I'm Professor Sunny, and thanks for joining me as I start asking questions. According to the US Food and Drug Administration, or FDA, people in the United States encounter mercury mainly through three routes: fish, where the mercury is in the form of methylmercury; some metal based dental fillings contain elemental mercury, the liquid metal form that slowly releases vapor; and a small number of certain vaccines in the form of ethylmercury, which is the breakdown product of a preservative called thimerosal. Separately, the U.S. Environmental Protection Agency, or EPA, estimates coal fired power plants have historically been the single largest human made source of exposure to mercury within the United States. In toxicology, the dose makes the poison, but the chemical form matters just as much as the amount. Methylmercury and ethylmercury, for example, differ by a single carbon in the group attached to the mercury atom. That small structural difference has impacts on how the body interacts with it, how long the compound stays in the blood, whether it reaches the brain, and how the body clears it. So let's take a look at these sources in turn to understand how most mercury in modern life enters the environment, starting with the one that actually accounts for the majority of a typical person's lifetime exposure: coal. Coal contains only a small amount of mercury per ton, but coal is burned in very large volumes, so the totals add up. By the United Nations Environment Programme's accounting, coal combustion is among the largest human made sources of atmospheric mercury, roughly a fifth of the global anthropogenic total. When coal burns, most of that mercury leaves the stack as elemental, inorganic mercury and disperses into the atmosphere. What happens next is what links the power plant to the dinner plate. The U.S. Geological Survey describes the pathway. Atmospheric deposition, such as through precipitation, is the main way mercury reaches most water bodies. And once it's there, bacteria convert the inorganic mercury into methylmercury. Methylmercury is absorbed easily and accumulates in tissue, so it concentrates as it moves up the food chain from plankton to small fish to larger fish. In top predators such as swordfish or large tuna, the methylmercury concentration can be more than 1,000,000 times higher, or 6 orders of magnitude greater, than in the surrounding water. For most people, the largest share of lifetime mercury exposure is dietary from eating fish. Methylmercury is an established developmental neurotoxin, and the evidence for that comes from some of the longest running population level studies in environmental health. One key data set comes from the Faroe Islands, where a traditional diet included meat from pilot whales, which is high in methylmercury. Researchers measured human exposure to mercury from umbilical cord blood at birth, and then tested the children as they grew. Higher prenatal methylmercury exposure was associated with measurable deficits in attention, memory, and fine motor speed, and these associations appeared at relatively low exposure levels. When the same children were retested at age 14, the associations were still present. A second data set points in a different direction, and the contrast provides important nuance that helps us understand the science. In the Seychelles, where people eat ocean fish almost daily, a comparable long-term cohort unexpectedly found no consistent association between prenatal methylmercury exposure and developmental outcomes. And in fact, on some measures, children with higher fish-based exposure perform slightly better. Two careful studies focusing on the same chemical, but two very different results. The explanation is not that one is simply wrong, it's that the mercury did not travel alone. In the Faroe Islands, it arrived inside pilot whale meat, which also carries other contaminants, especially PCBs or polychlorinated biphenyls. PCBs are toxic industrial contaminants that enter the food chain in a similar way. But in the Seychelles Islands, the methylmercury arrived with ocean fish, which supply nutrients associated with brain development, such as omega-3 fatty acids, selenium, and iodine. Environmental exposure never happens in isolation. So while a scientific study may think it's evaluating the effects of one chemical, it's tangled up with the millions of other things that are also present. When considering this, it's impossible to say from these studies what effect the methylmercury had on the people studied. Was it the PCBs causing the damage from the whale meat, not methylmercury? Were the omega 3s that powerful that they overcame any potential negative impact the methylmercury had? Was the methylmercury actually beneficial, an effect hidden by the presence of other toxins? We can't know. These studies don't actually tell us anything other than that toxicology is really, really complicated. Now, let's turn to ethylmercury. Thimerosal is a preservative used since the 1930s to prevent bacterial and fungal growth, and was commonly added to multi-use vaccine vials. Thimerosal is roughly half mercury by weight, and in the body it breaks down into ethylmercury. For many years, the safety assessment for thimerosal was based on the exposure limit set for methylmercury, the environmental form concentrated in fish. That raises the question worth asking for any safety calculation. Is that the right comparison? Are methylmercury and ethylmercury the same? When researchers measured ethylmercury directly in infants rather than inferring it from methylmercury, they found a compound with different behavior. In a study published in The Lancet, infants who received thimerosal-containing vaccines had blood mercury levels that stayed low and declined quickly, with an estimated blood half-life of about 7 days. And the mercury was eliminated largely through the stool rather than building up. A large follow-up study of 216 newborns and infants estimated a blood half-life of about 3.7 days, with levels returning to baseline within roughly a month. For comparison, methylmercury persists in an adult body with a half-life of around 50 days. A study in infant non-human primates compared the two forms directly, giving oral methylmercury to one group and injected thimerosal to another. The thimerosal-derived mercury cleared from the blood faster, and total mercury measured in the brain was roughly 3 times lower than in the methylmercury group. The authors concluded that methylmercury is not a suitable reference point for assessing the risk of thimerosal. The risks from ethylmercury have to be evaluated separately. It's worth noting the nuance that the literature itself points out. In laboratory studies on isolated cells, ethylmercury and methylmercury can show similar toxicity. The divergence happens when we start looking at real-life conditions in a living body, where ethylmercury is cleared rapidly at the organ level, not the cellular level. So the relevant point is not that one form is harmless and the other harmful in some absolute sense. It's that the two have different toxicokinetics. A risk estimate built on the wrong one will not be accurate. Thimerosal was largely removed from routine childhood vaccines in the United States in the early 2000s. The reasoning was precautionary. In 1999, as the total mercury an infant might receive across an expanding vaccine schedule was being added up, the U.S. Health Public Service asked manufacturers to reduce or eliminate thimerosal while further data were being gathered. From a manufacturing perspective, removing it is largely a question of packaging. A preservative's job is to keep a vial sterile when it's accessed more than once. A single-dose vial is used only once, so it doesn't require a preservative. And that's the format most high-income countries adopted. The preservative is most relevant in multi-dose vials, which allow many doses to be drawn from one container. Where a preservative is needed, alternatives do exist. Other compounds, including 2-phenoxyethanol and phenol, are used in some vaccines. A randomized controlled trial of 200 infants in the Gambia compared a multi-dose pneumococcal vaccine preserved with 2-phenoxyethanol against the standard single-dose version and found comparable safety and immune response, which is relevant to supplying vaccines in resource-limited settings. The alternatives also come with constraints. Thimerosal cannot be used in polio-containing vaccines because it inactivates the polio antigen, and multi-dose vials with an effective preservative are useful for distributing vaccine supply efficiently, including during surges in demand. These are the practical factors that weigh on either side of a formulation decision. This removal of thimerosal from U.S. vaccines has created a wonderful natural experiment, which we'll come back to in a moment. But first, a widely discussed claim is that thimerosal causes autism. Let's look at this claim because it'll help us understand what questions different study designs or scientific strategies are able to answer. One analysis reporting an association drew on the Vaccine Adverse Event Reporting System, or VAERS. VAERS is a passive reporting system. Reports can be submitted by anyone. They're not independently verified, and the system has no built-in comparison group. It's something a patient or a caregiver, like a parent, can submit on their own, and medical evidence isn't required. It's as close to being the comment section on YouTube videos as it is to being an electronic health record. Designs like this are well suited to flagging potential signals for further study, which is exactly what the system is designed to do, but they cannot, on their own, establish whether one thing causes another. Aside from allowing self-reporting without medical evidence, there's no controlled comparison to separate the proposed cause from everything else. If we take the self-reported data at face value, we still have no idea to what else these people were exposed. It's just like all of the unknowns surrounding the whale meat, while adding in the uncertainty of self-reported data. Matching the study design or scientific strategy to the question is the key step. A meta-analysis, or a combination of studies pooling more than 1.25 million children, found no association between autism and thimerosal, mercury, or vaccines generally, with an odds ratio essentially equal to 1.0. A separate meta-analysis examined 2 exposure types and reported no association between thimerosal and autism or ADHD, while finding an association for environmental mercury exposure. A reminder that the source and the chemical form of an exposure are not just important nuances, but essential aspects. There's also that natural experiment I mentioned. If thimerosal were a primary cause of autism, then removing it from vaccines between 1999 to 2001 would be expected to be followed by a decline in autism diagnoses. Researchers tracked autism prevalence in California and found that it continued to increase through and after the removal. Because a cause must precede its effects, removing the proposed cause and seeing no corresponding change is evidence against the proposed cause. A comprehensive reading of systematic reviews places thimerosal among the environmental factors found to be unrelated to autism risk. In June 2025, the Federal Advisory Committee on Immunization Practices recommended that children, pregnant women, and adults receive only single-dose, thimerosal-free formulations of the seasonal influenza vaccine. A few factual points provide context. About 96% of influenza vaccines administered in the United States during the 2024 to 2025 season were already thimerosal-free, and roughly 0.3% of pregnant women received a formulation containing it. The CDC's stated position is that available data show no harm from thimerosal in vaccines beyond minor reactions such as redness and swelling at the injection site, and no link to autism or neurodevelopmental delay has been identified. Among the considerations raised in the discussion was the role of preservatives in multi-dose vials for distributing supply, such as during surges in demand. These are the kinds of factual inputs that go into evaluating a formulation recommendation. Now, another question you may have been asking yourself: if coal plants emit the single largest source of mercury, and mercury is a neurotoxin, do areas near coal-fired power plants have higher rates of autism? Well, you're not the first. Several studies have asked exactly that, and they make a useful case for evaluating these different study designs. The most cited come from Texas. One compared county-level mercury released into the environment, taken from the EPA's toxic release inventory, against autism rates in school data, and reported that autism rates rose as reported mercury release rose. The amount of mercury emitted from coal-fired power plants directly correlated with autism rates. A follow-up examined distance to emitting sites and reported that autism prevalence was higher closer to industrial and power plant sources, roughly a few percent higher per 1,000 pounds of emissions, with the association weakening as distance increased. So the closer you are to a coal-fired power plant, and the more that power plant emits mercury, the higher the rate of autism will be. While those are real published associations, they are ecological studies. They compare group-level rates across places, not specific exposure levels and outcomes in the same individual. An ecological study design cannot establish that the children diagnosed with autism are the ones who are more exposed, which is a limitation the authors of the proximity study stated directly. At a basic level, the amount of mercury a facility reports releasing is not the same as the dose any single person actually inhales or absorbs. And just like with a study involving whale meat, confounders will exist at the ecological level. For example, places near industrial sources differ in many ways at once: population density, income, access to the clinics and services that produce an autism diagnosis, traffic levels, vegetation density, noise, light pollution, any number of variables we can't isolate could be contributing. While the association is there, the causation isn't scientifically established. Please, if you have an idea to design an environmental study that can disentangle all of these factors and more, post it in the comments. Studies designed at the individual level can offer a stronger look. A California study linked birth records to modeled air toxic levels and found autism risk elevated by roughly 50 to 70% in the highest exposure group for a set of metals that included mercury. A separate cohort-wide study using the Nurses' Health Study 2, which adjusted for family and neighborhood socioeconomic status, reported about twofold higher odds of autism in the highest versus the lowest quintile for several pollutants, mercury being among them. So is the question settled? Unfortunately, no. And the reason is a recurring one. The variable cannot be isolated. In each of these studies, mercury arrives bundled with diesel exhaust, lead, manganese, solvents, all from the same sources, among an unknowable number of other factors. And the studies are honest about those other pollutants and the difficulties involved. No design here separates Mercury's contribution from its co-emitted companions or environmental covariables, and few studies are able to track individual exposure levels to preemptively tie it directly to an individual's potential future autism diagnosis. Form matters too. Coal releases mostly elemental and inorganic mercury that's inhaled or deposited, different from the methylmercury associated with developmental deficits. And those well established methylmercury effects are on general neurodevelopment, not a demonstrated cause of autism specifically. A meta analysis reported an association between environmental mercury and autism while finding none for thimerosal, which is informative but still rests on correlational data, not causation. So, if we're concerned about ethylmercury from only rarely administered multi-dose vaccine vials, should we be more concerned about methylmercury present in fish? Should we be most concerned about elemental mercury from coal-fired power plants? The scientifically accurate summary is narrow. Some studies do report correlations between elemental mercury emissions and autism. The better studies measured effects at the individual level. But with the unfortunate limitations of the real world, none have been designed with enough controls to show direct links between individual exposure levels and a future autism diagnosis, completely independent of other exposures. Unfortunately, the exposure to one molecule in the environment, mercury in this case, cannot be separated from the other pollutants, environmental and social factors, and life events that might occur with it. It remains an open research question rather than a settled finding, and it may stay this way for a while. So, back to the opening question: a coal plant, a tuna, and a vaccine all involve mercury, but three different mercury compounds with different behaviors in the body. Methylmercury, concentrated in large fish, is a documented developmental neurotoxin studied over decades. Ethylmercury from thimerosal, almost completely absent from modern vaccines, is cleared from the body within days, and controlled studies have not found a link to autism. Elemental mercury from power plants and industrial sources is correlated through both distance and density with autism, but the association is practically impossible to disentangle from all of the other possible known and unknown factors. The through line isn't a verdict to adopt, it's a set of questions to ask. Which specific compound is it? What is the dose? How does the body absorb and clear it? What are the other sources we should be thinking about? How do we know which source is contributing the most? And can the study design actually support the conclusion being drawn from it? For toxicology, these questions work the same way regardless of what we're investigating. Aside from the complicated, nuanced nature of the available research, nothing I've said is intended as medical advice, let alone individualized advice on a personal level. I cannot provide that. For questions about diet during pregnancy or specific vaccines or any other medical related questions, the right source is a licensed clinician who knows your specific situation and can engage in evidence based, shared decision making in line with your health goals. So, until next time, please keep asking questions.